1, 2. 1, 2, test. Okay, good. Hi, good morning to everyone. Thank you for joining us back, for coming back for this second hybrid day. So today we've got two more sessions, session 3 and session 4. This morning, immunocells-based therapies with 5 overview and overview conferences and introductions and short talks. And there will be another question and answer session at about 9:50, 10 o'clock. Then a coffee break, and after that we'll have a round table. We'll have a keynote, important keynote with Liz Alter, head of AIS, the Missions of Health Innovation Agency, and then a lunch break at 12 noon for 2 hours. And then we'll come back for the last session, session 4 this afternoon, from 2 o'clock, with other short talks and questions and answers during the whole afternoon, with the participation of Philippe Ménacher or Alain Fischer, between many other people. To launch this session 3, I welcome with great pleasure Cécile Martina and Christophe Junot. Is he here or are you alone? Oh, you're alone. Great. Cécile Martina, to talk about National Research Program for Biotherapies and Bioproduction. Thank you.
Thank you. Thank you, Fabrice. Yeah, Christophe apologized, but he won't be here today. So first I would like to express my gratitude to the organizer for this meticulous planning of these 2 days of great change. Clearly yesterday, during the yesterday session on gene therapy, the richness of the exchanges illustrate the quality of this meeting, but also the need for the community to be gathered and to exchange, and also highlight on the next challenge that still that still remain regarding the development of biotherapy. So next slide. Oh. Okay. So it's a pleasure and it's an honor to be here and to kick off this session meeting, this second day, by having the opportunity to present you the national program called PEPR, Biotherapy and Bioproduction of Innovative Therapies. A program that has been officially launched in last December and that I've been co-piloting with Christophe Junot, of my colleague from CEA, and with the help of Sabrina Lizo from Inserm, which is a great, and I again thank her for the great job as a project manager. It is all the more important to present you today the PEPR, as this program is one of the first actions that have been launched thanks to the National Strategy for Accelerating Biotherapies and Bioproduction, and for which 2 days ago, in the same room, we were all celebrating the second anniversary. And during this anniversary, it was the opportunity to review the fact that in France, in 2020, they were clearly lagging behind the bioproduction of biotherapy. It was estimated that only 5 biotherapy were produced in France, against 21 in Germany and 12 in Italy, and that France was dependent for 95% on the imports for biotherapy production. And all this clearly highlights the fact that France urgently needed to regain its leadership position for biotherapy and bioproduction. So it is in this context that the national strategy of acceleration for biotherapy and bioproduction, that the PEPR has been launched. It's really important to keep in mind that this program is among one of the first steps of all the different initiatives that have been launched within this national strategy. And it's also important to keep in mind that the PEPR targets fundamental questions, fundamental research, with the aim of seeing the outcome of this fundamental research to be translated and to be converted into industrial or into clinical development, thanks to different tools like the integrator for applied research, but also Combio, and also for the industrialization, thanks to different programs such as the PIEC or France Polit. So more specifically, the aim of the PEPR is clearly to recreate and to foster the growth of a strong French ecosystem on biotherapy and bioproduction, to taking on different scientific challenge, and I will come back on the different areas that are tackled by this program, to again organize and structure the research community to promote the breakthrough, to strengthen the excellence of French research, and to regain the French as a leader position at the international level. And regarding the different areas that are tackled by this program, clearly these areas have been defined in function of the domain of the evolution of the technology. And the PEPR tackles cell therapy, tissue engineering, vesicle, extracellular vesicle, gene therapy, nucleic acid therapies, and antibodies. And we can summarize this program into 4 different axes with 2 transversal objectives. So each of these axes is about either cell therapy or tissue therapy, gene therapy, biotherapy and bioprocess with antibodies, and extracellular vesicle. And the 2 transversal objectives are clearly first to improve the development of current biotherapy and the deployment of new biotherapies, and also to prepare the France for the future innovative biotherapy and their mode of production. And again, and I thank the ANR, Lupenico from ANR for sharing this slide with us. This is again to insist on the fact that the PEPR is one tool among a large series of different tools that have been developed across the national strategy of acceleration. So again, the PEPR is here at the fundamental research level, and is combined with different initiatives that have been, most of them led by AENA or BPI France. So the PPR, it's a budget of 80 million dedicated for only this program for a duration of 7 years. As I mentioned, it's co-piloted by Inserm and CEA, and it's also in close collaboration with public, private, hospital research communities. It's structured on different actions. So one of the first action, and I will come back on that point, is the development of priority scientific project, 12, and I will show you which one are there, 3 per axis. I will also take some time to present you what we are doing actually with the upcoming call for proposals. So we are launching an AMI, so it's a call of manifestation of interest this year, and another call will be launched in 2025. And the PPR has also a mission in terms of animation and coordination project training. But I won't have time today to talk too much about this aspect. The general governance of the PPR, so I already mentioned the steering committee with Christophe Junot and myself, the project manager, top management from Inserm and CEA. We also benefit from the support of the International Strategic Committee that include different representatives from CEA, CNRS, Inserm, INRA, Pasteur Institute, with different invited members from Integrator Network, for example, also France Pulid, and I will show you that we are in close interaction with France Pulid, again, to have a feedback of what are the needs for industrialization in terms of biotherapy development, also patient associations. We also implemented an EAB, an International Scientific Advisory Board, that include up to now 6 members, and they are only international scientists, and the idea is to have the feedback of the international community in our field from what are the next challenges and how we need to structure ourselves. So, as I mentioned, one of the first actions of the PPR was to launch 12 priority projects, 3 per axis. All the projects here have been launched. The kickoff of each project have been already done. So you can see that for axis on cell and tissue therapy, one project led by Anis Benasser, it's about IPS, called IPS France. It's about the use and the development of IPS line in a GMP grade. Bioengineering Skin France by Nicolas Fortunel from CEA. eCondro by Farida Jouad from Montpellier. Gene therapy, so yesterday we had the example of Oumeya Ajali that lead a bioscale project for Target, Kuala AV for the qualification of production of AAV, and is led by Philippe Andral from IFCED. The Edito project from Thérèse Gredin from Nantes about gene editing. Axis 3 is about bioengineering antibodies with the Acredia project that is led by Bernard Mayer from CEA, RNAVac, and Chantal yesterday was here to mention and to discuss about the next challenge about the use of RNA for vaccine and for biotherapies. Thérapie Beta that is led by Anne Gali from ART TG. And the last axis, it's about the use of extracellular vesicles, so Stroma-F by Daniel Noël from Montpellier. So, as the name mentioned, it's the use of EV by Stroma Cell as a biotherapy tool. Bacter EV booster from Jean-Marc Châtel from Miscalis, and CARN, that is a project animated by Florence Gazot about the use of ARN. So each consortium, it's about 3 to 10 different partners. It's 4 years project, and these different consortia gather different research institutes, hospital, university, and Grande École. Okay, The next action for 2024, it's, as I mentioned, the launch of a call for manifestation of interest, AMI. So to design this call, we took advantage of different workshops or different discussions that we had, both with France Biolid, as I mentioned, but also with the International Scientific Advisory Board that we developed, that we installed, and with the Institutional Strategic Committee that clearly help us to give their feedback about what is still needed in terms of the development of new biotherapies. And what came up, it's the fact that it's still important to stick on the four different main words, keywords that define the PPR: gene therapy, cell therapy, bioengineering, extracellular vesicle, but that we still need to tackle different challenges in terms of new concepts and new biotherapies and advanced biomanufacturing. And for each of that, we need to implement and to introduce new discipline, new domain of research that were maybe not involved in the 12 projects that have been initially launched. So as an example, chemistry. And when I'm talking about chemistry, it's in the large sense, broad sense of the term. It's chemistry, organic chemistry for formulation, for example, artificial intelligence and data science. Clearly, we can hear everywhere about artificial intelligence. So how can we introduce data science and artificial intelligence for the development and the implementation of new biotherapies, and also engineering science? And again, the idea is clearly to enhance the research skill, to foster the design of innovative biotherapies and their bioproduction. So this is just to give you an idea of how will be the selection. So it will be so a budget of around 13 million euro will be dedicated for the IMI. We are thinking to fund around between 3 to 4 new projects, but we do not have a strict limit, and it will be really dependent of the pre-proposal that we will have. Again, the objective of this IMI is to foster the multidisciplinary, so again, to associate cell therapy with artificial intelligence or chemistry. And the official launch of this IMI will be during March of this year. So the selection process will be based on pre-proposal of consortia that will be evaluated both by the International Scientific Advisory Board, but also by the pilot of the PPR and the ESC. Then the selection of pre-proposal, the submission of consortia that will be evaluated by a specific committee that will be organized by INR, and then the selection of the project. And if you want to know more about PPR, about the launch of the IMI, I clearly advise you to follow us through our website. So I'm really proud to announce that our website has been launched yesterday. So we are just in time for my today presentation. So this is the address. And you can also follow us on the INR website, and because the PPR clearly benefit from the support of the INR for its organization. So thank you for your attention.
Thank you very much, Cécile. And of course, at the end of the morning, before the break, you could come back and answer a few questions with the others, okay? Thank you very much. Now let's welcome Tamas Shisha from Novartis for another overview conference.
Good morning, everyone. I am Tamas Shisha. It's an absolute pleasure to be back to Paris. I am from Novartis, translational medicine immunology, and I was asked to just give a short introduction to this session. And it's always amazing for me to come back to Paris. We've lived here with my family. Actually, my 2 daughters were born in Saint-Cloud, So part of my heart belongs here, even though we left 11 years ago. In this next session, we will talk about next generation of gene therapies, and this session specifically is going to be about how the use of CAR-T can go beyond the cancer therapy, how we can improve the delivery to solid tumors and applications beyond cancer. This introduction will be followed by short talks and a roundtable discussion. And my introduction is going to be about our own Novartis experience of going beyond cancer. And I'm going to present to you the Sentinel data from the first 3 subjects of this ongoing clinical trial in severe refractory lupus nephritis treated with CD19 CAR-T therapy. So the whole story really began with the cases presented by the group of Professor Schett in Erlangen. Many people have been thinking, obviously, about going beyond cancer with CAR-T therapy, but it was really, we in Novartis have been thinking about it, but there were many pros and cons, many risks, and doing the first step was an important one, and Professor Schett indeed made this first step, and the data was really quite spectacular, as you can see on this slide, with a dramatic improvement of the disease activity score, SLEDAI, on the left, complete depletion of the B cells, and not just peripheral B cells, but most likely, obviously, the deep B cell depletion in the tissues. Also, complete depletion of double-stranded DNA and interferon, while maintaining, as you see, limited impact on protective antibodies. So this was, so to speak, the trigger for us to also go on this endeavor and set up the study. So this is just a background slide, but I think I really summarized the contents already. So in terms of the methods, so this is an open label, a single-arm, multicenter study. Multicenter, we also have centers in France, several, one here in Paris in La Pitié, to assess safety, efficacy, and cellular kinetics, so YTB, in patients with severe refractory lupus. So I'm going to present this cohort of patients, the Sentinel cohort, that was dosed with 12.5 million cells. And we looked at CAR-T kinetics by qPCR and flow cytometry, and we looked at a number of other secondary endpoints, as you will see on the next slides. So here is the baseline disease characteristics of these first 3 Sentinel subjects. So you see that the patients were between 38 and 50 years, male, female. All of the patients received the same lymphodepleting chemotherapy before receiving YTB. They had a long disease duration of more than 10 years. All of the patients had high disease activity. You see the SLEDAI scores, 22, 14, and 12. And all of the patients had several major organ involvements. This is the safety profile at the time of the data cutoff for this Sentinel review. So you see all the adverse events. So this is patient 1, patient 2, and patient 3. In color, we have the grade 3 and the grade 4 events. So these are obviously the more severe ones. And as you can see, these are adverse events that one can really expect after the lymphodepleting chemotherapy. So these are all cytopenias, lymphopenia, neutropenia, leukopenia, lymphopenia. So these are reported in clinical trials as as they should be, but of course it was nothing that we did not expect, and the data monitoring committee had really no concerns, and neither did we. So on this slide you see the depletion of CD3 T cells with a pretty quick recovery. So this is the result, of course, of the lymphodepletion pre-treatment. And here the CD19 cells, which remain depleted throughout the period of time. So the longest follow-up was patient 1 until day 91. Clearly showing a distinct effect of the CAR-T therapy as compared to just the effect of the lymphodepleting pre-treatment. Here you see the efficacy. So SLEDAI improvement going from baseline values. So this line is day 1, so when patients received YTB, going down quite dramatically. So this patient 1 was at SLEDAI 0 actually at month 3. This is the physician's global assessment of disease activity, also showing remarkable improvements. And here you see double-stranded DNA and complement C3 and C4 improvements. We also plotted the efficacy improvement on top of the data that was reported by Professor Schett's group in Erlangen. So the gray lines are the data from Erlangen, and the colored lines are the patients ongoing in our own study. We just wanted to see whether the effect and the effect size is in line with what we would expect based on the Erlangen data, and we were comforted to see that it's very much in line. And so this preliminary data really prompted us to go on and to continue the study. And currently we already have treated 10 patients, so since this data cut off. So it's going fast, and more data is to come. And we will be proud to share the data with you. Next opportunity will be in Bruges, in Belgium, in March at the European Lupus Conference. And then in June at the EULAR, we also plan to present an update from this study. And with that, I would like to thank you for your attention.
Thank you very much for this great introduction. Very short and quick and very impressive, Tamas, Shisha. So you will come back to on stage for a few questions and answers. Now we'll have several short talks. Let me precise to all presenters who will come, if you can try to speak as close as possible to the mics so as to be well heard by everyone. And even if you show, if you can try to stick to the mics, it's better. I'm sorry, it's just for the comfort of everyone and for video recording too. So now let's welcome Suman Mitra. Okay, Suman Mitra. You'll talk about optimizing adoptive cellular therapies in physico-chemical tumor environments for superior anti-cancer efficacy without systemic toxicity. And you're from Inserm. Thank you.
So thank you, the organizer, for the opportunity. So we're interested in understanding how cytokine work, and this is really critical because this is the messenger how immune cells talk to each other. And many of the time, multiple cell types can respond to the same cytokine, yet have a very distinct function, and sometimes multiple cell types and this pleiotropic action. So the studies has been always how to decouple the cytokine pleiotropy in order to make cytokine more cell type specific. And this is important because most of the immune disease you can relate to the cytokines. Now, the last 20, 30 years, we've been studying cytokines in very optimal conditions. So in our lab, we have incubator, and we have very optimal conditions in terms of temperature, the pH, the oxygen levels, and nutrient-rich media. But very often, in the tissues, that's not the case, because the temperature varies. Arthritis, you can have a very different temperature, in particular inflamed tissues. The pH can really drop to something around pH 5.5 to 6. And oxygen levels could drop from 20% to 1%. And also the nutrient could be different in different tissues. So for example, you can have much more potassium, sodium ions hanging in the tumor tissues. So I think this is really important to really study cytokine or cellular activity in the context of those chemical environment. So we are taking an approach of one cytokine and one environment at a time. So today I'm going to discuss about acidic pH, because this is a hallmark of the tumor because of the glycolytic activity of the tumor tissues. So we rationalize because immune cells, when they enter this pH environment, they really shut down the production of cytokines. And because the cytokine receptor interaction happens in the extracellular space, the chemistry of the extracellular space can really influence the ligand receptor interaction. So we looked at varieties of cytokine, and we can say that at least 40 to 50% of the cytokine we have looked into has really impede activity under acidic extracellular environment. So we looked at IL-2 and we solved the crystal structure and looking into the acidic environment, how it can happen. We found that there is a histidine-arginine salt bridge in this critical interaction of IL-2's high affinity IL-2 alpha interaction. And as you know, the histidine has a tendency to get protonated. So when you have a H+ protonation, that cannot interact anymore with the arginine residue. So you have a repulsion. And that leads to this impede activity. Now looking into the many other cytokines, we can really find this salt bridge interaction in multiple variety of cytokine. So in the lab, we use yeast display system to engineer proteins. So we rationalize if we can engineer a cytokine, or in this case IL-2, that could be more active in the acidic environment and less active in the systemic organs, we can perhaps switch its activity where it's needed. So just to give an example, an IL-2 is one of the first immunotherapy that we know of. And high-dose IL-2 therapy works, but it's only few patients respond. So we could show that either using a lactic acid deficient tumor, or in this case, sodium bicarbonate, if you use sodium bicarbonate, you could see that IL-2 therapy works much better. And this is direct effect, because we could look at the direct proximal signaling of this IL-2, and you could appreciate that in the tumor with the pH, low pH, the IL-2 induced STAT5 is much lower. But in case of when you neutralize the tumor with the sodium bicarbonate, the STAT5 is really higher. So IL-2 therapy is highly sensitive to the acidic environment, and tumor unable to acidify their environment are really sensitive to the IL-2 therapy. So IL-2 variants that is highly thriving in acidic environment could have a really profound effect on the target while minimizing the toxic effect. So using yeast display, we created a molecule called Switch2. And Switch2 has this unique property that it has a very low activity in the systemic organs, bind to the receptor, very low affinity, but it have a full-blown activity in the acidic tumor. Shown here, the crystal structure of normal IL-2, and you can see the salt bridge interaction. Again, this histidine is there, it gets protonated, it have a repulsion. By our Switch2, what we've identified that we have an additional of different histidine, H35. So now, this gets protonated and can interact with the aspartic acid. So there is this positive-negative interaction that Switch2 is enabling Switch2 to active in the acidic environment. So we think that this strategy could really be useful in terms of how we think about cellular activity and how we can modify cytokines in terms of making them more active in tumor environment. So we have done a lot of different assays to show that this is really the case in terms of looking at in vivo signaling, but just showing the tumor data and showing how IL-2 and Switch2 could differ. So looking at MC38 model, we could show that we can at least cure 40% of the mice. And these are also inducing antigen-specific T cells. But one of the major effects of high-dose IL-2 therapy is toxicity. And by reducing the toxicity in the systemic organ and looking at here by edema and the lung weight, and looking into the pulmonary syndrome, you could see that they switch to have a minimal toxicity in the lung, which is very heavily oxygenated tissues. So just by changing how IL-2 interact in the extracellular space, depending on the pH, we could get this effect. I think our study also changed the 20 years of dogma of IL-2 therapy, that if you target IL-2 to CD8 cells or NK cells, as opposed to Treg cells, you could get better effect. And as you know, many of the trials, including some of the pharma companies here, have tried to make IL-2 more active to the CD8 and NK cells, and they have a huge toxic effect. I think what this study can show that, what we really don't realize that high affinity IL receptor alpha is also expressed by the antigen-specific T cells, probably 10 times to 100 times more than the Treg cells. So we miss out when you're only targeting the dimeric alpha beta gamma receptors. So by switching the activity to the acidic environment, we could really target antigen-specific T cells, and thereby also, depending on the pH, can reduce their systemic toxicity. It also enables us to have a less sink effect and have a much more accumulation of the product onside the tumor, so thereby enabling us to have a higher concentration. So going forward, we are also looking into multiple different chemical factors in cytokines, and also hypoxia, redox examples, and we're also looking to see how this cytokine can really boost the activity of the cellular therapy, because that's what even if you CAR T cells, if you put in a hypoxic and acidic region, it does not make cytokine, doesn't kill the tumor. So we are hoping that by enabling CAR T cells to have this pH switchable, or we're also designing synthetic receptor of cytokine, we can make those much more effective in the hypoxic and tumor niches. This is probably the most important slides. So this is led by Silvia Gaggero in the lab with multiple collaboration, and I thank you for your attention.
Thank you. Thank you. Now let's welcome Emmanuel Donnadieu from CNRS, and you're going to talk about predicting efficacy and toxicity of CAR T cells using an ex vivo human model, and then you'll take part to the roundtable at the end too. Thank you.
Thank you very much, Fabrice. Hello, everyone. And I'd like also to thank the organizers for inviting me today and to present our technologies actually to test the efficacy and potential toxicity of CAR T cells. So, as you know, there are a number of challenges in the field of CAR T cells, although some patients respond extremely well to this strategy. Up to now, a majority of cancer patients is refractory to such a therapy, especially in solid tumors. So really highlighting the presence of resistant mechanism that one needs to identify in order to overcome them and propose more powerful solution. And among the different resistant mechanisms, the suppressive tumor microenvironment, as we've heard from Suman, Mitra, is really important. Another challenge, which is related also to CAR T cells, is the huge number of immune cell products which have been developed. I'm sorry, the slide is a little bit crowded, but here you could see at the center the different cells which have received the agreement, and all around you have this constellation of cells which are cell products which are either in preclinical development, phase 1 or phase 2. It could be autologous CAR T cells, allogeneic products, and immune cells equipped with a CAR. I'm sure we hear about other cell types which are very important, also NK cells, gamma delta, mate cells, and others. And the important question is, in which model can we test this novel T-cell product? Because we know that the current models are not the good one. So definitely we need more relevant models in order to test efficacy and also potential toxicities. So we at Inserm, Institut Cochin, we have been involved in the development of a new model. It's an organotypic model. It's based on fixed slices made from human primary tumors. So we have ongoing collaborations with clinicians at the Hôpital Cochin that provide us with fragments of fresh tumors. Could be, for example, non-small cell lung tumors. And this is a protocol. I won't go into the detail, but basically we cut the tissue with a vibratum. We make fixed slices really to maintain quasi-intact the tumor microenvironment. And on top of fresh slices, nothing is fixed. We add the T cells, could be T cells purified from the same tumors, the T cells, or it could be some T cells which have been genetically engineered to express a CAR. The T cells infiltrate the tissue is rinsed, immunostain to look at for the tumor cells, or stromal cells, stromal component, and then we use analytical technologies, including imaging microscopy. So we could look at, and this is some of the example, we could look at the distribution and migration of CAR T cells. So you have an example of CAR T cells in green, in human lung tumor slide. The tumor cells, it's very compartmentalized organ, as you could see. The tumor cells are in white, the stroma is in red. It's one image every 20 seconds for 20 minutes. And you could see that most of the CAR T cells are actually not in regions where we would like the CAR T to be in contact with tumor cells, but really excluded from the sequestered into the stroma. We are also able to look at the activity of CAR T cells, focusing on the calcium response. We use specific calcium dye for that, and I'll just show you an example. When the CAR T cells they get into contact with their target, you could see that they present a flash of calcium that we analyze using automated softwares. We could keep the explants for up to 2 days, allowing us to look at the killing abilities of CAR T cell, their cytotoxicity, and again, we use fluorescent dye. And in that pictures, you could see that the CAR T cells in red, they are associated in regions where you have many apoptotic cells. So we have used this technology over the years to really identify a number of obstacles that prevent T cells and CAR T cells to perform their job. We've, for example, identified a subtype of macrophages, and I think I have some movies there, a subtype of macrophages present in human tumors that really sequester T cells in the stroma. You could see there by doing video microscopy, but the macrophages in red, they form long-lived interaction with the T cells, really impeding T cells from contacting tumor cells. We also shown that the extracellular matrix, which is a hallmark of growing tumors, also dictates the way T cells migrate. As you could see there, the T cells, they migrate along the fibers, which are parallel to the tumor stroma interface. We've also recently shown that the lack of adhesion molecules at the surface of the T cells really prevent the T cells and the CAR T cells to form an immune synapse. So now that we have identified a number of obstacles that I've just described, our goal is really try to design some innovative strategies actually to boost the efficacy of T cells, of CAR T cells. So we have a number of strategies, developed a number of strategies. We could increase the migration of T cells by, for example, boosting the migration and the metabolism of T cells. We could obviously reprogram the macrophages, for example, neutralizing TGF-beta, targeting the extracellular matrix. Also, we have designed a number of different strategies, and we're also working on an epigenetic factor, and we have some, I think, nice data showing that we knock out this epigenetic factor, CX6A5, we could increase the persistence and the expansion of CAR T cells. So in terms of perspective, I think the platform that we have developed is very amenable for evaluating the effect of novel cellular immunotherapy. We're also doing, we plan to screen for therapeutic molecules able to restore T cell activities within tumors and within an intact tumor microenvironment. Use a slice assay also for testing the toxicity. And if we are interested in CAR T toxicity, please feel free to reach out to me. Of course, for conclusion and as a wrap-up, we have developed a relevant preclinical patient-derived tumor model to develop a number of innovative targeting strategies to improve CAR T cell efficacy. We have a strong collaboration with clinicians for obtaining fresh tumor biopsies. And we are a part of a number of national, but also international consortium, the French National Consortium UNITC, supported by INCAR and Cancer Grand Challenges on CAR T cells and pediatric solid tumors and T2Evolve also. And I think, yeah, so the work has been done at the Institut Cochin, the team Cancer and Immune Response, and I'd like to thank all of our collaborators, the clinicians, the academic, but also industrial partners, and you for your attention.
Thank you very much. Thank you, Emmanuel, and great team at Institut Cochin indeed. Thanks a lot. And we'll talk about it again a bit later. Now, Marie-Caroline Dieu-Nosjean from Inserm about manipulation of tertiary lymphoid structures at the 3rd generation of immunotherapy.
Thank you very much for your kind invitation. I'm very pleased to be here and to talk about one of our research programs in tertiary lymphoid structure that I co-lead with my colleague Jean-Luc Theillot present here today. So as you know, the tumor microenvironment is a very highly complex milieu and highly heterogeneous regarding different components of the tumors and also regarding the immune infiltrate. And so many years ago, we were very lucky to see that when you look at the immune microenvironment, you can see that in some patients, in some tumors, you can see lymphoid cells that can aggregate together. And here you can see the highest level of organizations that we name tertiary lymphoid structures, TLS, with a T cell area here adjacent to a B cell area. And you can see that all the cells that we used to see in the lymph node, for instance, are present here in TLS in the tumor of some patients. We were also very exciting to see that when you take into consideration the presence of this TLS in lung cancer patient, for instance here, you can see that more you have, you can see that more you have mature TLS with both T and B cell area, better is the outcome of the patient, even in a very advanced stage of the disease here. And so you can see that the median survival was not reached compared to patients having low density of mature TLS. So now this observation has been observed in many other solid tumors, for instance, breast cancer, even triple negative breast cancer, colorectal cancer, renal cell carcinoma, even in glioblastoma. More recently, in melanoma, in CCRCC and sarcoma, and more recently, and as illustrated here in advanced metastatic NSCLC patients, you can see that the density of TLS can also predict the response to immune checkpoint blockade. As illustrated here, you can see that patients having high density of TLS signatures, better response to anti-PD-L1, the survival is better compared to the other group or patient treated with docetaxel. Interestingly, in this publication published by Italiano in Bordeaux, Institut Bergonié, they have shown in sarcoma patients that if you pre-select patients according to the density of TLS, you can have a better response to anti-PD-L1. Indeed, here you have all-comers patients where you can see that unfortunately anti-PD-1 has no clear effect, not enough. But if you pre-select TLS high sarcoma patient, the objective response rate is very better, higher than in these unselected patients. So knowing that TLS is a powerful prognostic and predictive biomarker in cancer, in many types of cancers, our objective is to try to induce TLS in order to improve the survival of the patients. So first, to try to find the way to induce using cells or molecules in patients in order to modify the tumor microenvironment, and then to try to convert cold tumor to hot tumors, and then in combination with ICIs, try to overcome resistance to this strategy. So how to induce TLS? There is some publications in the literature talking about different molecules that have been tested in a preclinical murine model, where you have different members of a TNF superfamily. You have many chemokines and cytokines. So indeed, it works in mice, but you have also side effects, and it is not selected to specific to TLS. These molecules are known to be involved in other biological functions. Different cells have been positively associated with the presence of TLS, and others, when you deplete, for instance, Tregs, you can also improve, induce, or boost the formation of TLS. In the lab, we were very interested by the innate lymphoid cells, type 3, so ILC3, Why? Because they can do most of the job of these populations. They can uptake antigens, present antigens to CD40 cells. They can secrete cytokines, such as interferon gamma. And they can also kill tumor cells by a direct effect in a trail-dependent manner. So we were very interested by this population. And in addition, when you take into consideration what's happened during the embryogenesis, a subset of IC3 named LTI cells for lymphoid tissue inducer cells, so it's a subset of IC3, they are very important for the induction of the lymphoid organogenesis. So our hypothesis is that in adult, the IC3 may play more or less the same job and can induce the formation of TLS under inflammatory situation. So it's what we are currently testing. So we have set up a mouse model with lung inflammation. You can see that it is very interesting to see that the kinetic of the IC3 recruitment in the inflamed lung parallel the kinetic of lymphoid aggregates and TLS induction in the lung of the mice. So you have exactly the same kinetic. The optimal timing is around day 10 here. And at day 10, you can detect the presence of IC3 within the TLS in the lung of the mice. More importantly, when you sort the different immune cells infiltrating the inflamed lung, and you look by single-cell RNA-seq the expression of many genes, you can see that only IC3 here express many genes related to lymphoid organogenesis. So we hypothesize that these cells may have the ability to induce TLS in this model. So the perspective is to clearly demonstrate in vivo that the presence of IC3 is a very critical and a powerful TLS inducer cells. We are testing now the ability of IC3 also to participate in the TLS-based immunotherapy. And we think that using this strategy, we can induce a long-lasting anti-tumor immune responses following the induction of TLS within the tumor. And the end will be to test this combination with other therapies such as immune checkpoint. So in summary, what we are currently working is to identify different TLS inducers. I have talked about IC3, but we have other projects regarding new molecules that we are testing in vitro and in vivo in order to try to convert TLS low patients into TLS high patients. And the next step will be to combine this TLS inducer with immune checkpoint in order to boost the ongoing anti-tumor immune response taking place within the TLS. So thank you very much for your attention.
Thank you, Marie-Caroline. And to end these first short talks before having a session of questions and answers, now Josh Lourenço and Michael Saitakis to talk about a new class of tumor targets for next generation cancer immunotherapies and to present the startup Nemotherapeutics.
Hello, everyone. First, I would like to thank all the organizers for the opportunity to be here. We as Nemotherapeutics, we are a French biotech. So yes, I should. This is not this one. So we are a French biotech based in Paris, created in December 2018. Since the inception, we have raised around 80 million euros in equity and granted about $25 million euros, sorry, in non-dilutive funding, most of it from BPI France and from France 2030 programs. The company is based on technologies from Institut Curie on Sébastien Amigorena's lab, who is actually CSO and co-founder or scientific co-founder of the company. We are working on differentiation target discovery platform to develop immunotherapies and in particular cell therapies covered by 23 patents. The labs are located in Paris intramuros with 40 people working there. Most of them, I think more of 70% of them are scientists under the supervision of an experienced executive team. And on this, I will leave the floor to my colleague Michael to present the science.
Thank you, George. So at NEMO, So at NEMO, we're trying to address all the challenges related to immunotherapy of solid tumors, and I would like to thank also Professor Donnadieu for the brief introduction of the CAR-Ts. So our approach is really to generate best-in-class targets and best-in-class immunotherapeutics. So for this, we have developed two different but very interconnected platforms. One that is called e-antigen discovery platform. E-antigen discovery platform, and one cell therapy platform for immuno-oncology. So our best-in-class discovery engine now is looking for splicing antigens, so that we're trying to find new proteins that come out from this splicing that are either derived from alternative splicing or non-canonical. What does that mean? It means that in the tumors, actually, there is epigenetic alterations that result in the expression of transposable elements or other not annotated parts of the genome, and this result in specific splicing events between canonical exons, so exons that belong to regular proteins. These proteins, then we discover this and we call them e-antigens from epigenetic, and these results in their expression and translation. And from our pipeline, we have two types of these antigens. First, our regular proteins can be processed and degraded and then be presented as peptide MHCs, but also we have discovered that these proteins can also splice and be expressed as part of specific transmembrane proteins that can be directly targeted by immunotherapeutics. And we're developing this platform with the goal to generate, for these transmembrane antigens, some adoptive cell therapies and T-cell engagers, and same for peptide MHCs, adoptive T-cell therapies, T-cell engagers, but also vaccines that can be either semi-personalized or off-the-shelf. In parallel, we have our CAR-T platform that is addressing the T-cell exhaustion and persistence in vivo, and basically, what we're using is a new type of CAR that was developed in the lab of Michel Sadlan, one of our other co-founders, and an epigenetic reprogramming based on the disruption of this epigenetic modulator, SUV39H1. In this case, the T cells, they are more resistant to exhaustion, they persist more in vivo, and they have stemness characteristics. This results in this case, they outperform regular CAR-Ts in all the phases of the immune response, which means initial expansion, primary response, persistence in vivo, and protection from tumor relapses. So all these have been, we have published most of these. It was published last year in Science Immunology, and this was recently published in Cancer Discovery last month. So if you have more information, we forgot to put the papers, but please, you can feel free to look for them. So our key message from this presentation is that we're using these splicing events that are not canonical and are only happening in tumor cells, but what we're trying to achieve is to find them and really target them with immunotherapeutics. And we have two types of targets. The these transmembrane proteins that are really special and specific for the tumors, but also these peptides that can be targeted by vaccines. And we have proven now in vitro and in vivo that these antigens are druggable by immunotherapies, including T-cell engagers and cell therapy. And our pipeline also includes these off-the-shelf antigen-specific approaches, TCEs, cell therapy, and vaccines for high unmet medical need tumor indications. And our approach not only for specific indications, but we have discovered through our pipeline that there are other specific pan-cancer antigens that can be targeted with our approaches. So we currently have this full pipeline for our path to clinical development, and we're pursuing also these different kind of targets in an agnostic way to try to find the optimal immunotherapeutic approach. So we have several transmembrane antigens, several peptide MHCs, including for different indications, so specifically leukemia, ovarian, and some pan-cancer. And currently also we're working with our strong collaboration, we have a strong collaboration with Institut Curie, and we are pursuing two different clinical trials that have been supported by BPI and ANR. And one is for a rare indication, peripheral CNS lymphoma, targeting CD19, and one is for a solid tumor basket trial with targeting mesothelin, using our proprietary technologies for epigenetic reprogramming. So I would like to thank you for this opportunity, and happy for having questions.
You can stay on stage, yes. So now it's time for questions and answers, and all the previous speakers can go back to the stage. Tamas, Shisha, Cécile, Martina first, then Suman, Mitra, Emmanuel Donnadieu, Marie-Caroline, Dieu-Nosjean. You can take a seat, normally just enough. Aha, one is missing. So who is? Okay, that's kind of you. Thank you. You can sit just at the end. Thank you. So it's time for your questions. We have plenty of time. So who wants to start first? Mark, maybe.
And I have plenty of questions.
Oh, plenty of questions. Good start. Thank you.
Regarding the PPR, so I was wondering what was the strategy to involve private players in your strategy, at which level? And although this is, I know, TRL, a low TRL, I think that even though the private players could have a say on what you are doing and prioritizing.
No, thank you for the question. Clearly, it's an important point. So as I mentioned, the PPR is really dedicated for fundamental research. So we're not supposed to clearly have a direct interaction with industrial, but we need to have a discussion back and forth with private companies to see exactly the expectation, the challenge, the technical challenge, the scientific challenge. And then to develop. So we need to be in constant interaction. It's why we have a strong collaboration with France Biolide, for example, and a strong exchange with France Biolide. It can be also thanks to this type of workshop to see exactly the expectation at the industrial level. So we need to keep in touch all the time.
Do you want to carry it? Oh, no, there's one question, and then I'll go back to you. Okay, please.
Hello. I wanted to ask a question to Emmanuel Donnadieu with his nice system. I was wondering, hello, I'm Philippe Benaroche. So there is a lot of debate to really identify why CAR T-cell fail against solid tumors, among which there is a lack of metabolites, there are induction of Tregs, there is problem of access to the solid tumor due to the extracellular matrix. I was wondering whether with your system of slices, you can manipulate your system by playing with the extracellular medium and so on, which could be the main cause of the difficulty of CAR T cell to treat solid tumor? Because it seems when you discuss with CAR T cell specialists that they disagree on what is the actual main reason for their failure to penetrate into the solid tumor and indeed do their job there.
Thank you, Philippe. Can I have 1 hour?
Two.
It's so complex why CAR T cells are not able to control solid tumors. Well, as you said, it's very complex. Multiple steps are probably altered. As you said, probably the entry of CAR T cells into the tumors is dysfunctional, the tumor microenvironment very hostile. Another aspect is the target. Do we have the good target? And we heard about the importance of the target during the last talk. Probably no. I mean, probably the target that we are probably not the good one. Most of them are also expressed by healthy tissues, causing some kind of toxicities. So we definitely need to have better targets. So I mean, again, the tumor microenvironment, for the fact that most T cells are dysfunctional, the patient have been highly pretreated usually, and so we know that the phenotype of the T cells is very particular, very dysfunctional. So that's also why we probably need to think about other T cell cell product, allogenic product probably. So I don't know if I replied to your question, but as I said, many steps are altered in solid tumors.
Maybe if other speakers want to add something to that, if you want. Get your own opinion on that topic now? Okay, no problem. Yes, please.
I have a question regarding the Novartis trial. So if I am right, you are using the T-charge system with, for people in this room, a system to generate fast CAR T cells that are supposed to be less exhausted, more fit, and persist longer in cancer. And in cancer, this is what you want to have, because if your CAR T cells do not persist, you have recurrence. But in autoimmunity, what we know from the Erlangen trial is that the B-cell recovery is rapid, and that the lupus, notably, and the other conditions that were evaluated, does not recur. So I was wondering what was the rationale for Novartis to try to use super fit CAR T cell that would persist, because what you want is B-cell recovery, so you don't want them too long. And did you see also a B-cell recovery in your trial, or is it too early?
Thank you for the question, and it's a very relevant one. First of all, to your last question, we have seen B-cell recovery since the data that was shown here. So in Bruges, we will show that data as well. You are right, we don't know how long we need the B-cell depletion to persist, but that's not the only reason T-charge makes sense. First of all, the time from leukapheresis to the time that the patient can receive the treatment is much shorter with the T-charge, so that's really a key aspect. And it's also easier to manufacture. I was at the manufacturing site, and classical CAR T is almost a, excuse me, almost a floor of people working, dressed up like they could go to the Mars. It's very, very work heavy. The T-charge is just a room full of people doing the same. So it makes it faster, it makes it easier. And from the patient's perspective, the key is that it's faster back to the patient.
Yes.
Yes, hello. Can you, same question to Tamas or Shisha. Can you elaborate on why, what CAR T cells can do that rituximab could not do in these patients?
Well, so most likely there is a deep B-cell depletion, which goes way beyond what peripheral B-cell depletion rituximab can do. And the most obvious conclusion and the most obvious proof of the difference is the difference that we see in terms of clinical efficacy. Because B-cell depletion, 100% peripheral B-cell depletion we see with rituximab, but when we look at the data in lupus and when we look at the data in lupus nephritis, we do not see anything that resembles the data that was presented by Professor Schett
or the data that we generated in our first couple of subjects in lupus and lupus nephritis.
Okay, yes.
Yes, I've got a question for Cécile Martina regarding the PEPR. You mentioned that you are going to attract some other competencies, including chemistry, and so the question is, how are you going to attract chemists in your program, and are you planning to launch some actions?
The main action will be the communication, and clearly it's again, it's an important point. We need to be attractive. We need to communicate on these domains that were not involved yet. So we already started, but locally, and the idea is to expand. And we were waiting for the website because it will be also a way to communicate. And it's also why I'm here today. It's to start the communication and to start to sensitize about the fact that we need these other domains.
Yes.
Yeah. A question to Caroline. You mentioned that the density of TLS can predict the efficacy of the immunotherapy. Can you elaborate regarding the quality of this TLS? And also my second question is, can CAR T cells modulate the density of this TLS?
So there is a huge discussion right now regarding the state of development of TLS. So there is at least 3 different states: lymphoid aggregates that should not be functional, immature TLS, and mature TLS, as I show you. So we don't know exactly whether immature TLS is unfunctional or incomplete in terms of development, because we have to think about the dynamic of these structures. So at the T time, we saw that, but we don't know what's happened 2 weeks after and so on. So it's difficult to say more about that, but for sure you need to have a good organization and a good segregation with T and B cells, and most probably you need both area, as I show you, in order to have a better outcome. Most probably there is lots of data suggesting that you need a good coordination between the T and B cell responses. And most probably maybe they target the same antigens. So we have seen that in at least lung cancer patients. And regarding CAR T cells, I don't think that there is data exactly regarding your point, but there is a nice publications of one publication where the authors develop light CAR T cells. So light was one of the molecules that I mentioned, and indeed in their models they are able in mice and humanized mice, NHE mice, they are able to induce TLS and to have a better efficacy of the CAR T regarding the tumors. So it's not exactly your question, but there is some development regarding the modification of CAR T cells in order to, in addition to the function of the CAR T, to try to induce TLS and to combine both advantage of this strategy.
Do you want to add something? Yep.
I just want to have a follow-up question for Marie-Caroline. The idea of that having more TLS, more T and B cells, and Andy Mean has shown that you have very inflamed tumors and melanoma, and they do very poorly to the checkpoint blockade. And if you add JAK1 inhibitor, and they seems to work much better response to the anti-PD-1. So I just want your thoughts about in this highly inflamed tumor, TLS, and to what extent they are good, and to what extent they could be also, resistant to the therapy.
Yeah, yes, for sure. Some people try to see whether there is a relationship between the immunogenicity of the tumors and the efficacy of the prognostic value of TLS, let's say. And there is no relationship. So it's not related to the oncogenic drivers, immunogenicity, because even in tumors, for instance, CC or CC, which is with low immunogenicity, TLS are also associated with a good outcome. So I think it's more complicated situation. And what was the second part of your question?
you block JAK1 inhibitor or you suppress?
Yes, I don't know. But most probably if you combine different strategies, I don't know for JAK1, but for instance, when you block Tregs, when you deplete Tregs, you have a better, more TLS, you have a boost of TLS and a better protection of the mice. So for sure, if you can manage the balance, you induce TLS and you try to inhibit the immunosuppressive mechanisms, it should be better. And ICI is the best example to date.
Okay, thank you. Yes, Mark.
Yes, I have one question because Emmanuel mentioned briefly that you were contemplating modulation of some epigenetic modulators. And so you mentioned one that was different from the one from Pneumotherapeutics, so SUV39H1. So I was just wondering whether between RINF and this other one, whether they were complementary, whether they were redundant, and because basically in both instances they seem to modulate the survival of CAR T cells.
Yeah, so the one that I was mentioning, the RIMP or CX6F5 discovered by Frédéric Pandino in a team, is actually a platform for TET2. So we know that TET2 is also very important epigenetic factors, also controlling the differentiation, the expansion of CAR T cell. Now the question is whether there is also a link between the THUV epigenetic factors and RIMF. That is something I don't know. But it's quite complex. There are a number of different epigenetic factors which have been shown also to, when they are knockout, to promote the expansion and proliferation of T cells. There are also this other DNMT3A, I think, by Sanjud, Gochak's lab, showing that it's also very important. So it's also very complex. You could really tackle so many different factors, intracellular factors, transcription factors, epigenetic factors, really having drastic effect on T cells, their differentiation and expansion.
And another issue, because so you described nicely in your tumor model this exclusion of CAR T cells from the tumors. So I was wondering, what was your strategy to create a kind of rationale for particular combination approaches? For instance, typically, I was wondering whether you have tested immuno-oncolytic viruses?
No, yeah, I mean, there are different strategies, of course, to overcome these exclusion barriers. The extracellular is one of these, and we have a number of programs, actually, with Laurence Gazo in the audience, to target the extracellular matrix. You could do that in different ways. You could prevent the reticulation of collagen into big fibers. We've done that in preclinical models. You could try to degrade the extracellular matrix. So the idea that we had at sometimes was to produce a collagenase by CAR T cells that would be able the T cells to migrate more efficiently. They degrade the extracellular matrix. Also, nanoparticles could be very attractive. We have also a project with Florence to, by doing hypothermia to degrade the extracellular matrix. Also, magnetic nanoparticles with some magnet, you could try to retain the CAR T cell specific. So again, this is very complex. I think it is to be fully tested in different preclinical models. One model is probably not enough. Yeah. So, and I haven't talked about oncolytic viruses, about also producing chemokines that would attract CAR T cells at very specific location, but there are many opportunities.
Michael, you want to add something?
Okay, if I may add on the epigenetic part, so I didn't describe a lot, but I think there are also in different pathways, there might be some interactions. So the RINF, if I'm not mistaken, it's controlling TET expression, which is part of the DNA methylation path, while SUV39H1 is more on the heterochromatin part, so it's controlling chromatin accessibility. So now it's actually a very exciting field in T-cell biology, working on the epigenetics and trying to control the differentiation.
Agree.
Right, yes.
I have a question for Suman. So congratulations for your talk. It was brilliant, the idea to modulate the effect of IL-2 depending on the acidity of the milieu. But as you know, and you alluded to that, if you give low-dose IL-2, you activate the Treg, so it's supposed to be a regulatory mechanism. And then if you give high-dose IL-2, you activate more the memory B cells, notably, that express the beta-gamma IL-2 receptor. So I was wondering if you engineer an IL-2 mutein that is less effective at normal pH, don't you think it could be the equivalent of a low-dose IL-2 and therefore maybe elicit more Treg expansion that would not occur in the tumor, but in, let's say, draining lymph node, and they could traffic to the tumor. So in your MC38 experiments, for instance, what did you see of the Treg? Is it a stupid question, or can you have an effect on that?
Thank you, Olga. I think that, to what extent Tregs is really critical in IL-2 non-responsiveness, still not very clear, So we do see some degree of Treg cells, but it is more abundant of antigen-specific T cells, in ratio to the Treg expansion. And, we can also argue that a little bit of Treg cells may be better for reducing the toxicity of IL-2. So, I think what we are alluding to here that, as you said, like most of the time we're targeting CD8 cells versus the Treg cells. And what that happened, that you can also induce a lot of NK cells in a systemic organ, leading to, very severe toxicity. So by changing this pH adaptability of IL-2, we could still have this low-grade, low IL-2 signaling systematically, and perhaps that is also critical in terms of those maintenance of memory cells, but also have a high dose IL-2 effect on the tumor. So you can still increase some amount of Treg cells, but there is much lower in compared to the antigen-specific CD8 cell, because they express probably 10-fold more CD25 than a Treg cells.
Okay, yes, please.
Yes, Michel Cognier, just one question to Marie-Caroline and Emmanuel, and that's about potential correlation between presence of TLS, which might be open structure, with lymphoid channels, and the exclusion of some tumors. So do you see some correlation between both elements? More TLS when you have less exclusion?
I think you should go ahead, Marie-Caroline, because we didn't look very precisely at the link between TLS and T-cell exclusion.
Yes, so for sure, you can have a huge T-cell infiltration and not so much TLS. This situation, unfortunately, occurs. And for instance, CD8 T cells are no more related to the survival or prognostic for the patient. But when you have TLS, you have a huge immune infiltration. So it means that there is a close relationship between the TLS and the microenvironment. And most probably, TLS can shape its own microenvironment. So when you have TLS, you have plenty of immune cells, T, B, DCs, macrophages, and maybe not the same macrophages as the one that you can see on cold tumor, or when you have an immune desert, by definition, there is no immune cell. So yes, there is a relationship, and yes, the idea is that maybe TLS can shape its own microenvironment. So I don't know, when you perform migration on the tumor slices, if you can see some difference regarding the migration of cells, depending on whether you have TLS or not on the
We'd love to do that. We never really try to correlate the migration and the localization of T cells in relation to TLS, but I think we should look at that, absolutely.
Maybe chemokines are not the same, so maybe we have to go further into the analysis of the chemokines. So it's something that we will try to do with the spatial transcriptomic analysis on different kind of solid tumors.
Last question, maybe? Yes?
I have a question probably more for Emmanuel Donnadieu. It's regarding the activity of CAR T-cell in solid tumors. We know now there is a lot of heterogeneity of endothelial cells in tumor. And are there any data correlate the T-cell activity with certain heterogeneity of endothelial cells in tumor?
Thank you for the question. I mean, not to my knowledge, but I'm thinking at the same time, and there are many correlations, obviously, with the, as we discussed, with the structure of the tumor microenvironment, but studies showing the relationship between CAR-T efficacy and the nature on the endothelial cells. Now, this is a very important point, obviously, in solid tumors, because this is the entry step for CAR-T cells. So, I mean, we know, not in the field of CAR-T cells, but for immune checkpoint inhibitors and others, but the vessels are very abnormal. They don't produce enough chemokine, that the adhesion molecule is also altered in the level and really impeding T cells from the extravasation step. So it would make sense, a lot of sense to look at. But I have to say that people are really focused in the tumor microenvironment, cytokine, chemokines, but not much on vessels. So we should really explore that aspect too. So thank you again for the question.
Okay, no more question? Oh, last one. Okay, Mark, let you. We have a bit some time.
Marie-Caroline, so you mentioned the opportunity to develop ILC3-based immunotherapies. So how would you contemplate the design of this kind of therapies? For instance, typically, do you have evidence that, for instance, after large-scale expansion, the ILC3 cells keep their properties, and when you administer them systemically, would they migrate just like activated lymphocyte to tumor sites?
There is two answers. The first one is yes, in mice it works. When you inject by IV ILC3, they can go back to the lung, for instance, because we're mainly focusing our studies in the lung. And the other option would be to try to attract ILC3 within the tumors. So we're working on that field also.
Thank you very much. Thanks to all of you. So coffee break, and please come back at 10:50. Thank you. do you mind sitting here? Thank you. Just waiting for Emmanuel. Emmanuel, you're not here yet? Not sure? Okay, we're waiting for our last panelist, and then we start. Ah, okay, Emmanuel. Thank you. We're going to launch the round table. So we're going to continue talking and thinking about all the incredible things and problems, issues that were told earlier in the morning with a great panel and with also the participation of industry, which is always very important and interesting to see the bridges between the two and, of course, the strategies. But to connect to what we've just said before, I'd like to start again with you, Emmanuel, and it will launch the rest of the discussion and, of course, questions and answers after our first exchange. You talked about many important issues and things, but maybe you could focus on toxicity. You just said a few words about it, and I think that it's one of the main issues that you face. Neurotoxicity, cytokinic storms, and others. Can you tell us more?
Well, thank you for the question. Is it working, the microphone?
Normally, yes. Just try it.
Oh, thank you. No.
Okay.
Oh, no,
thank
it's
you.
fine. Thank you. Yeah, no, of course, this is a very important issue, the toxicities. We know that there are, as you said, different kinds of toxicities, those mediated by an exaggerated immune response, a cytokine release syndrome. But this one, I think the clinicians have learned how to manage the cytokine release syndrome, IL-6 production, and neutralizing IL-6 and its effect. There are also the neurotoxicity, which is also, it was not predicted at all using the current preclinical model, the mouse model that scientists were using. We missed totally the neurotoxicities.
How can you explain it? We have an idea?
Because simply the current model that we are using testing the efficacy and potential toxicity are not the good one. For the moment, yes, that's right. For the moment, we are using immune-deficient mice transplanted with human tumors and infused with human CAR-T. And in these animals, we missed totally both the CRS, but also the neurotoxicity. And so clinicians were quite surprised initially to see such severe toxicities in some patients. And I have to say that I think, I'm not a clinician, but in terms of mechanism underlying responsible for this neurotoxicity, still a lot to learn. Is it due, for example, to the infiltration of CAR-T into the brain, to also the diffusion of inflammatory cytokines to the brain, or eventually there was that paper from UPenn showing that CD19 could be also expressed by a very small fraction of brain cells, the neural cells, which are covering the vessel. So eventually.
So it could be targeted.
That's right. So again, still a lot to learn. And yeah, I mean, of course, we need more predictive models.
So you work on humanized mice? That's right.
Humanized mice could be one model. We are working at Cochin Inserm on another model. Actually, I presented the model with tumors this morning, slices made from human tumors. But we are also developing the same model also for a European IMI consortium, T2Evolve, with healthy tissues, slices from healthy tissues. And so we apply CAR-T cells onto healthy tissues, slices from healthy tissues, and we looked at potential activation of the CAR-T and destruction of healthy cells. Because forgot to say that another important toxicity is the so-called on-target of tumor toxicities. The fact that when the target is expressed also by healthy cells, even at very, very low level, tiny levels, of course the CAR T cells, they don't make the distinction between the tumors and the healthy cells. It is what's happened actually with the commercial CD19 CAR T. We have a total destruction of the healthy B cells. But when the target is expressed by vital organs like the lung or the heart, then it could have very, very severe side effects. So we definitely need to have a better model in order to predict such toxicities.
So a great challenge among others, and of course one of the most important is to have people more responding. You have 20 to 30% of patients who are responding, but all the others don't respond. So that's a very important issue. And Santé Koundari, you are Senior Director for Gilead, Head of External Scientific Collaboration in Medical Affairs, Oncology and Cell Therapy. So, of course, you address this problem. You were one of the first companies to work on CAR T cells. I think it was 2007. It was early, very early. And now you want to address this problem, especially which is one of the most important issues, on solid tumors. You talked about it a lot this morning. Solid tumors is very important to reach today, and you work on application multiple myeloma and others, but solid tumors is one of your most important targets.
Yeah, I would say that we are continuing to work always in hematological malignancies. That will continue to be our, let's say, main goal for the near future, but we have some assets starting in solid tumors, especially in brain tumors, thanks to a collaboration we have with Immunity and Penn University. So we would like to address and to target glioblastoma and neuroblastoma, also with a focus on children. It's really early stage in terms of assets, but is one of our, let's say, future things to look and to see in the future. With all the, I would say, challenges we discussed before, because solid tumors is really different for the liquid, I would say, cancers. But again, we hope that first, having the right target, that could be one of the issues we already discussed, could be one of the things that I think from a sponsor and company perspective, we want to see if the right one. And then, yes, finger crossed that we are working hardly on that. Let's see how we will work.
Okay, so we'll go back to the strategy and maybe autologous and allogeneic approaches, which are also another issue that you all share. But on the same topic, Vigo van Tondelu, sorry. I was just saying, but the photo is not the good one. Okay, sorry, I've got to make a round. Sorry. So, Vigo, Director External Innovation and Benelux for Johnson & Johnson. And you also addressed the question of solid tumor, which is a very important issue for you too.
Absolutely.
No, it should be working now.
Yeah. One. Yeah,
okay.
Yeah, sure. No, and just as Santé said, the main focus, of course, of Johnson & Johnson is still to develop CAR Ts for the hematological malignancies. But of course, we're also looking very intensively to the field of solid tumors. As we are aware of the bottlenecks and obstacles that are still out there. And I think they can only be tackled by what I would call synthetic engineering of the CARs in order to arm them with potencies and features that could withstand this tumor microenvironment and able to invade the tumor and exert their function. So I think we're not there yet, for sure, for solid tumors. There's a big road ahead for us, and I don't think we can solve all the problems, but I think if we can combine efforts and we can collaborate, I think that would be the goal for solid tumor CAR T cells, and use the knowledge that come from academia and see how we can apply that knowledge in a translational way to make ultimately to come up with better treatment for patients. So, yeah.
All right, and so you have different strategies, and you think about some specific issues. For example, current therapies are focused on alpha beta T cells, but you say there could be an interest to focus on gamma delta T cells.
Right. Next to engineering, let's say the classical CAR T cells, I think we should be open to other possibilities of not only T cells, but also looking at other cells. But even within the T cell population, I think there's a lot to be learned on which T cell subset we should aim to arm with a CAR T and not just take the whole population of the T cells. We have indeed a population called gamma delta T cells, which are known for their intrinsic cytotoxic effect. So if we could arm these T cells with CAR Ts, with a CAR T construct, I mean, that would make sense, of course, to make even a better or a more potent CAR T.
Another issue that we'll, of course, share together is that you'd like to move from oncology, which is our main focus, to autoimmune disease.
I think many people are now looking into the possibility of moving from oncology to other domains as severe autoimmune diseases, as we heard this morning from Novartis, to use CAR T cells as a way to deplete the autoreactive B cells. So indeed, I mean, it's a very young field in its infancy, of course, and we need, of course, to really outweigh the risks of CAR T and the manufacturing challenges, of course, for CAR T in that area. But I think good strides are being made to also use these CAR T cells in autoimmune disease.
All right. So now, Alessandro, I'm not mistaken, Alessandro Crotta. So your global program leads cellular therapy development at BMS. You've already got 2 products approved in that field, Abecma CAR T and anti-BCMA, that's right. And you work on multiple myeloma too. And you also want to, well, you focus on cancer, but you also, as we said, focus on immune disease.
Yeah, correct. So at BMS, of course, we have deep knowledge on what CAR T can do in hematological disease like lymphomas and multiple myeloma. But now we are focusing also, as many others, on autoimmune disease. So we have ongoing trials in different autoimmune disease. That's the beginning, because of course we heard this morning about the data in lupus. We are looking at other disease in autoimmune that have autoimmune background, like also neurological disease. So it's a quite large area that can be investigated where there's a potential for CAR T cells.
All right. The question of the production of these CAR T cells and the use of patient cells, you say that maybe there could be an interesting issue with iPS cells?
Yeah, I think we already see the limitations that we may have with autologous CAR T cells, because it's a very complex process for each patient. So we need to find ways that make more efficient and scalable the production of these cell therapies for patients' populations that are larger and larger. And of course, autoimmune disease, it's a first example of a very large patient population, much more larger than what we are now treating in hematological disease like lymphoma and multiple myeloma. And so for that reason, we look at other platforms that can be used, specifically allogeneic cells that can be donor-derived, but also iPSC that are an interesting platform to be used for expanding the production from one single source for multiple batches.
Okay, so it leads us to that very important question, autologous versus allogenic platforms. And there is another way. Karine Rossignol, you're CEO of Start Immune. You were one of the co-founders of Imagine Foundation with Professor Alain Fischer that we'll listen to this afternoon, namely. So you're a great specialist of the question for many years, and you propose something that could answer that problematic, which is pro-T cells, which would avoid all the situations we want.
Yes, indeed. So we are developing a new T cell subtype, the T cell progenitors. We can differentiate in only 7 days ex vivo hematopoietic stem cells into what we call T cell progenitors, meaning T cells which have not been educated on a thymus yet. And the interest of that is that we can have a kind of the best of the two worlds, meaning we can use HSCs coming from an healthy donor, differentiate them ex vivo, and then infuse them to the patient to be treated. And his own thymus is going to differentiate those cells into a fully polyclonal new T-cell compartment. And those cells could express CAR-T. And in that case, we could have CAR-T, which could have a much longer lasting effect, because indeed educated by the own patient thymus.
Okay, what's the stage now? Where are you?
So we have validating the concept and the rationale in hematology post-transplant. All the data are pointing in the right direction. We have demonstrated that we can rebuild a fully functional T-cell compartment in severe combined immunodeficient babies. We are at early stage, this is only 10 patients included, but demonstrating the same in adult leukemic patients.
Right.
So we have a project which is still in preclinic on off-the-shelf CAR-Pro T-cell, which is very exciting.
Great. Well, Emmanuel, what do you think of this approach? Allogenic logics and this way?
No, I mean, as I said, I mean, conventional T-cells, alpha beta T-cells, they have many advantages. You could produce them in large numbers, but also they have some drawbacks, limitations, especially for an allogenic perspective. If you want to use an allogenic T-cells, you will have to manipulate genetically that T-cell. That could be very complicated. So instead of doing that, of course, using other cell types such as NK cells, gamma delta, NKT cells or other cells or iPSCs would present a very important advantage, of course. It makes sense, and I'm sure we'll hear a lot about developing some immune cells equipped with a CAR. Right. And also including, and I know that we have also experts in the audience, some innate immune cells equipped with a CAR. It could be monocyte, could be macrophages, it could be B cells. So we are lucky we have all experts in the room on these aspects.
Okay. Right. On this issue, on this autologous versus allogenic approach, what's your point?
Well, I think that, of course, everyone now is especially in the companies, I mean, and Johnson & Johnson in particular, we are focusing on these autologous products. But of course, we see these enormous manufacturing challenges to supply all the CAR T cells for those patients in demands. So we need to work on a solution in the background, of course, to be able to treat every patient that needs that therapy. So, and I think then allogeneic, and even if I can go one step further, even in vivo CAR T cells could be one of the solutions. But of course, I mean, much work needs to be done there, and it goes without saying that off-the-shelf CAR T strategies do make sense from a logistic point of view. To have enough batches and CAR T cells ready for the patient's demands. But again, we face some immunogenicity problems with the allogeneic CAR T cells. We can engineer them, or we can re-educate the T cells again. So I think there are some solutions on the horizon.
Okay. Do you look at it, something?
Yeah, we are working. We are working on allo as well. I think we have some assets in early stage as well. And obviously, personally, I don't see auto versus allo as a fighting. I see how the asset can be complementary and also helping some of the issues of the others. Obviously, on the paper, allo, it represents a big hope, I would say, bypassing, problem of having the asset and the treatment ready for every patient in needs, et cetera, et cetera. Still, I think we have some issues, and we just heard that there are some solution of it. Cell gene editing is helping to avoid some of the big problem for GVHD, for example, that we can have for the allo as well. To me, and this is my personal thoughts, is also to verify how the scale-up of the allo will be in the future, because the industrialization of the asset then is very important. As company, we are working, let's say, how to improve the autologous asset, and just to reduce the timing, I would say, the product to the patient. So we are working to automations and to work to produce and to restrict the time for the auto. Again, it's complementary, is absolutely a very good hope. Still some challenges, I would say, but we are close to recover and to save, I would say, these challenges.
Yeah.
Maybe also what is important is the quality and the profile of the cells. So if you take autologous and you use T cell of a patient which has undergone a lot of treatment lines, those T cells are going to be damaged. So they're going to have a hard time to provide, even with a CAR, the efficacy expected. If you think about it, IPS is definitely the way forward. I think we are not there yet for T-cell application, because again, a T-cell is supposed to stay in the body during 7 to 10 years. So there is obviously tumorigenicity, safety to be challenged there.
Related to the APS.
Exactly. So at Smart Immune, we do think that going first by using T-cell progenitor is one step in the right direction. But of course, on scalability, IPS would be definitely the ground, and we are all pointing in that direction. But again, quality of the cell is key to overcome exhaustion hurdles. Sorry.
All right.
I fully agree with what has been said, but maybe another aspect which is important when we talk about allogenicity, is the cost. Of course, it will decrease, and I think I'm sure we will discuss about the cost of the product. Yeah, but having an allogenic product will, of course, decrease the cost of the treatment.
So a treatment is around about $300,000, I think. It depends, of course, on treatments, but the prices that we can see here or there, you've got two products. And of course, there's the problem of targeting the patients. If you only get 20 to 30% of responding patients, how do you target them? Do you make kind of a companion test, or do you use imagery to try to follow the distribution of the CAR-Ts? How do you select your patients, or do you know how it works in clinic?
Yeah, so this is a $1 million question, I think, that everyone has on which are the right patients to treat with cell therapy. And of course, we are, as of now, I think with the experience that we have with the different products already on the market and where there's more experience even in the real world setting, I don't think that there's any signal for now that provide a clear answer to this question. There's still a lot of work that can be done and is ongoing to clearly identify those patients that benefit best out of the available treatments. And of course, especially when you move, you want to give this kind of therapy in earlier lines, this is more and more important because there are standard treatment that works very well for some patients, but not for others. So be able to identify those patients who are not getting benefit and may have benefit with a cellular therapy, that's the main point of the work that we are doing right now, and try to leverage on the existing data, on the amount of data that everyone of us is collecting through the studies.
Yes, yeah.
Yeah, no, I would agree with Alessandro. I think that now the aim is, especially if these assets are moving in earlier therapies, where, as Alessandro correctly said, there are standard therapy, maybe the focus is to, let's say, to select the high-risk patients, where there are some genetic and molecular, I would say, signatures that are not responding to the standard therapy, and this could be in one space when so innovative therapies can be applied, I would say. And on the other side, I think that now in Europe, with the big registries, and I think France is representing a very good examples how the registries can deliver a lot of information. With the national registries you have here, but Europe is following, is also helping, let's say, to identify with big data, maybe cluster of patients that can be, let's say, very good responders versus others. So I think that the match between, and the technology can help, between big data analysis and molecular signatures could be also a field and the future field how to work all together. I think private, so sponsors, industries, and academic centers.
Okay, so there are some very modern approaches that you talked about this morning. The idea of add magnetic nanoparticles to follow CAR-Ts into the distribution with RMI. Is it something that could be imagined in the future?
Of course, we need to validate that using preclinical models. So, I mean, the idea, and we have an ongoing collaboration with Laurence Gazot also, is to use magnetic nanoparticles and to load the CAR-T cells with these magnetic nanoparticles, inject them into an animal bearing a tumor, and then using MRI to follow the biodistribution. I mean, it's very important where the CAR-T cells go.
That's right.
We know that in patients, the first organ where the T cells are going it's not the tumors, it's the lung. They are sequestered into the lung for 2 days. So it could also raise very important issues about toxicities in the lung. And the other aspect, of course, since these nanoparticles are magnetic, then you could add magnetic fields or a magnet in order to retain or attract
to the tumors.
Because we know, and we heard this morning about the different obstacles preventing, at least in solid tumors, really preventing the CAR-T cells to home correctly, to infiltrate correctly. This is really a major obstacle. So having some strategies able to attract T cells would make a lot of sense.
Why not injecting directly the tumor?
It has been done, of course. I mean, for example, for brain tumors, even, I mean, I could refer to a publication done by a colleague at Stanford, or Imaginer, Christian Michael, treating some children with glioma, some kind of very aggressive glioma, and they infuse the CAR-T cells in the brain, and they actually evidence very promising result, although not enough to cure these poor children, but really very encouraging results.
So do you work on that kind of strategy to inject directly the tumors?
Well, as of now, we are not focusing on that because that's not an approach that is feasible in many diseases where you have one lesion that you can easily target and inject your cells. So the beauty of the cell therapy is really to leverage on the natural tendency of your immune system to fight against cells that can be distributed everywhere in your organism. So this is the main point of cell therapy, that you give just an injection and then the cells knows where to go and then they are activated.
When it works.
But of course, the main, I agree with, especially with solid tumors, the infiltration of the tumor is one of the main difficulties and barriers that we are facing. So how we can prevent that and overcome that issue, and how we can engineer these cells to overcome that issue.
Okay.
Yeah,
yes.
I agree, and Johnson & Johnson has recently been embarking on something called interventional oncology, meaning that we would like to treat the tumor at the site of the tumor. And of course, for cell therapy, I mean, it's still a long way, but we're envisioning how to, instead of using the systemic delivery route of drugs, how we can inject and deliver the drugs really on-site using robotic platforms that, of course, we use from our other sector, Johnson & Johnson MedTech. So we're really joining forces now with the MedTech sector in order to come up with innovative solutions to deliver intratumoral.
I think artificial intelligence must be helpful to help design.
Absolutely, and that is throughout the whole portfolio, of course, that AI is making its introduction in not only in drug discovery, but also in various parts of data sciences and analysis of big data and real-world data. So absolutely.
Great, thank you. Do you have questions for panelists? I see Mark is ready, but if you have, okay, you want to start? Yeah, take your mic, thank you. Yes, normally. Can you hear? No, okay. I think the mic, try again. I think, is it connected? Are mics connected on the desks?
It works, no?
Yeah.
Okay.
Thank you.
Many thanks for the very interesting discussion. I'd like to ask a question about dose reduction and potential for cost reduction related to that. We've seen a number of studies, including the T-charge protocol data that was presented earlier today. Also, the studies from UPenn where decreasing dosage have been shown as effective, if not in certain cases more effective, given reduced time of expansion and arguably less exhaustion of the cells. So I'd like to ask the question this way to the panelists. Do you see any lower limit to the dosage that could be achieved and still be effective? And in that way, to reduce the cost of the treatment?
Very good question. Who wants to answer?
Maybe.
Yes,
again,
yeah.
Using the right type of cell with the right capacity to expand could be one solution. If you can use the right type of T cell and you get the right percentage of TSCM, meaning cells who are really fit, numerous, able to expand, we may participate to both improve efficacy and diminish cost and doses. The other solution, I'm sure, it's about scaling and manufacturing, and I'm sure there is a lot of hypotheses and scenario and methodologies investigated on the stage.
Yeah, I think it's part, I would say, of the solution. It's not really let's say, the solution in terms of lowering the cost, because I think it's also costs are related to other variables other than, the dosage and the administration. Sometimes you need to have, to give the possibility to the patients and, to be treated. And then on the manufacturing side, that you need to be sure that you are developing and having the right cells to be administered. So the cost is also related to some, I would say, possibility that you have a second bag, for example, or others able to be administered to the patient. Is part of the solution, is not totally related to the cost. My point of view is that with therapies that are one shot, theoretically, always you can avoid other type of treatment. So the cost containment is when you have the possibility to manage very well in a health system where companies and institutions are aligned. Maybe there are other collateral costs that you can maintain. It's not only the cost of the product itself. And maybe the intervention and the alignment between companies and public is also to see how to harmonize those treatments on the field, I would say. So it's a complex questions where there is not one right, I would say, answer, at least my personal opinion.
Okay, yeah. Alessandro?
I think the reduction of the timing of the cell expansion, it may not be the solution for each of the products, because for the available products, we know that the dose count because it's important, and there's a dose-dependent response. So may not be applicable for all the cell therapy that are developed. For some of them, yes. And of course, there's a lot of work to focus on how we can improve the in vivo expansion. And that's what they are leveraging some of these platforms. That, of course, it's important. Overall, on the cost, I don't think that the main driver of increasing the complexity and the cost of the production is like 2 or 3 additional days in incubation to just grow more cells. It's all the complexity around the production system. But of course, it's definitely something that, as you can hear, someone is already looking at, and we are also looking at how we can reduce, because, for example, even in our product that we are developing in autoimmune disease, we are taking a new platform that is reducing the time of production. So those are all points that we are taking, but may not be the response for everything.
Thank you. Other questions? Yes.
The question for Karin. Amazing technology around thymus. I was wondering, you're still talking about adult thymus, like when you're infusing them, they're going to whatever thymus you have left over, and whether cytokines such as interleukin-7 could be useful, because there are thymic implantation that has already been FDA approved, but they failed to develop naive T cells. So I was wondering, what are your thoughts around that?
It's a very good question. Thank you. So we have demonstrated both in vivo, but we have also some first data in men, showing that there is a crosstalk, actually, between the pro T cell and the thymic epithelium. And you're right, IL-7 is very involved there, but it seems like our cells can increase thymic epithelium activity, and especially medulla, which is in charge of educating and differentiating fully the pro T cell into effective T cell. So the first data we have seems to show that we are able to provide resentymic immigrant even in elderly patients, meaning that there's still sometimes tissue, and there could be a way to wake up thymic activity. That's what we aim to demonstrate. And that was actually the reason why the Bill and Melinda Gates Investment Strategic Fund did invest in our company. They are not, they did support an oncology phase 1-2 in adult leukemia, but they are not really interested in oncology. They are really intrigued on whether or not our technology can reverse thymic involution.
And you talk about investment. Bill and Melinda Gates Foundation investing in your company, that's right?
Yeah.
So that's noticeable. Thank you. Okay, other questions? Yes, please. Can you turn on the mic, please?
Oh, yeah, sorry. I have a question from Emmanuel. Actually, you talk about the magnetic tracking of the CAR cells and using the mouse model to do that. So my question is like, is there any way that you use the data from the mouse model that you use for toxicity to find the relevance of the outcome, that how it could be in the human? Because you told me that the model is not like efficient enough to compare the toxicity in humans. So how you actually, what would be the readout relevance to human when you use this tracking of the CAR cells to understand the infiltration of them in the human?
Well, I think these are two different aspects, the tracking, the biodistribution. And again, also, if we're able to attract T cells to the tumors, we may actually prevent the T cells to home too much time into the lung or other healthy organs. So it would make sense also to developing such approaches. And concerning the other models to test for toxicities, then I will go directly to human ex vivo 3D models, especially for the on target of tumor toxicities, having a tissue that is susceptible to express the target and test for CAR-T activation and potential cytotoxicity, as we are doing actually, testing a large number of different CAR-T for the T2Evolve consortium.
Okay, thank you. Other questions? Mark?
I would like to go back to the approach of CAR-T in an allogeneic setting. So clearly this is a very promising approach, many challenges naturally. So first one, avoid alloreactivity of CAR-T, so you have to knock down the endogenous receptor. Avoid rejection of allo CAR-T, and then what could be the most promising approach? I remember several early cellular approach in infectious disease, where people used to select cells expressing commonly expressed HLAs. You have the possibility to knock down HLAs. It's completely doable now, but basically then you will trigger rejection by NK cells. So what would be your most promising strategy to optimally use CAR-T?
Great.
Okay, thank you very much. It's a fascinating question and not a $1 billion question, as you would say. We do believe that, of course, what has been done today in allogeneic CAR-T is amazing, and the addition of the cells and of the HLA or TCR does allow to get those cells into the body of the patient and avoid GVHD. We do think that the Thymus can do this job maybe more safely first and as efficiently. At the maximum, we would like to avoid to edit those cells. So we are developing with our R&D team, and they are very talented. Dr. Olivier Negret is around, an algorithm to choose among healthy donors, homozygote donors, and the more relevant HLA able to be used in an off-the-shelf biobank, so that 20 biobanks could cover somewhere around 60% of one population. That would be, to us, our goal, meaning it's a biomaking approach.
It's your purpose? That's what you'd like to develop yourself?
That's what we are developing.
You're
yes.
developing right now, okay. Good.
I think this is the main challenge for allogeneic CAR T, because we want to have a product that can survive and be active like autologous CAR T. I don't think that what we have right now in the allogeneic field is the response. There's much more work that needs to be done. And I think you touch on an important point on how we can avoid the rejection. So and these require multiple engineering. So the aspect of, and the difficulties of entering into the allogeneic field is really how many additional engineering you need to modify your cells to avoid GVHD, to make these cells persistent and get the right target. And so there are options already, also, for example, expressing HLA-E to avoid the NK rejection and so on, but of course increase the complexity of the engineering and how we can simplify that part of the engineering and make it sure that we are producing a safe product, and how we face all the regulatory hurdles that we have with multiple engineering of a single cell.
And one point, if I can add, is how much we need to engineer those cells. In the reality, would reduce the cost at the end, because we start that we say, oh, maybe potentially this can be a cost reduction.
Yeah.
We are seeing that on the paper, yes, is an ideal aspirational, but at the end, if we need to go deeply in engineering a lot those cells,
it will cost a lot.
At the end, the cost, I don't know how much will be the difference at the end. We need to evaluate.
So it means that you don't look at the strategy?
No, this is what we are looking also at the end, but at the end, we need to see what currently we have. We are working, with also academic partners in terms to see what could be the best solutions. But
you let them do the hard job and then you see afterwards?
Together, I would say. Together, this is part of my job, is to make those kind of deal and partnership. But again, I think that as scientists and clinicians and companies, we need to understand that aspirationally, yes, this is a very good science to be, and there are solutions, and there are very good solutions, but how much we can intervene, and then the cost is not the reality, the lower we can have. It's also something to evaluate.
And the exhaustion of the cell, because they don't like to be edited. So again, I'm not saying that progenitors are the solution to everything. It's another way to try to tackle the problem, and we will need to collaborate to find the right way to make it efficient in the body. But what is true is that there is cost for all those engineering steps, but there is also the fact that a T cell needs an HLA and a TCR to live 7 to 10 years in a human body. If they don't have it, they don't know how to interfere with their environment. So we need to find the right way to tackle cost, exhaustion, safety, and work all together, each of us bringing one piece to the jigsaw to make it happen for the patient.
Yeah, I think the key is collaboration in this, absolutely. And
you'll agree on that, I think. You'll agree on that point, yeah.
Yeah, absolutely, and actually it's my job as external innovation, to look for partners that are agile and nimble to tackle these sub-questions of the big questions, of course. And indeed, we are also putting a lot of effort in this allogeneic approach. And again, the engineering is very complex, because you want them to be immune stealth, but you still want them, of course, to be immune reactive at the same time. So there's a lot of engineering costs that comes with it. And again, and I bring it up again, we should also look at how do we want to make these T cells. when we do talk about T cells, I mean, that is a variety of cells that still can be, let's say, we can pinpoint towards alpha beta or gamma delta, which might have already features that we don't have to engineer upfront. So.
Yes, typically, well, for gamma delta T cells, naturally. But, well, you have two ways to address this. The first one is typically gamma delta cells, like NK cells, might have NK receptors that would make them more prone to detect stress-induced ligands and so. But also, and I think that this has been addressed by some companies, but not that much, the possibility to transduce gamma delta T cell receptors that are directed against conserved antigen that are upregulated on a wide variety of solid tumors. And I think that this is really an interesting opportunity because these receptors seemingly show limited toxicity. They show strong no alloreactivity, strong reactivity against a broad array of tumor cells. And I think that this is really a track that could be...
Absolutely, fully agree.
Yeah, and we are looking, for example, at that. But as we were saying, I think there are so many things to look at the same time, and that's why partnership are extremely important. So that's what we are doing at BMS, that we have. We are trying to create and be at the center of an ecosystem of innovation and try to create strategic partnership with different actors from the academia to biotech that have the most recent cutting-edge technology that can give us multiple strategy to address all these questions that are a lot of questions that need to be addressed at the same time.
So, B2B sessions are here for that. So in a few minutes now, from now, during 2 hours, you'll get occasion to discuss of these points. Thank you very much to all of you for your participation. Thank you. Great, great, indeed. So before getting this time for B2B sessions and lunch break, now let's welcome Liz Alter to talk about the missions of the Health Innovation Agency, a very important and precise keynote. Liz Alter.
First of all, I'm very delighted to be here today with you. Thank you for the invitation and the opportunity to talk to you today after this fascinating roundtable. The discussions and exchanges were very rich. So thank you very much to the panelists. For decades, France has been one of the birthplace of cell and gene therapies, thanks to the collaboration between all the actors of the ecosystem. First, thanks to patients, of course, with the huge involvement of AFM Téléthon, who invested since the 19th 800 million euros to develop gene therapies, as you know, that changed lives of patients affected by orphan diseases. Thanks to physicians who pushed back the limits of science when Professor Fischer succeeded with the first gene therapies for bubble babies. Thanks to worldwide recognized academic research teams, of which we can mention Inserm, Imagine, Geneton, APHP. Thanks to the collaboration between all these talents, France records huge successes in the field of cell and gene therapies. Our researchers are behind developments of some of the most groundbreaking therapies, like Zolgensma for spinal muscular atrophy, Strimvelis for immunodeficiency, or Zynteglo for beta thalassemia. France hosts the biggest platform for AAV vectors manufacturing, Hypothesy. France is the second country in Europe in terms of clinical trials for gene therapies. And 20 early-stage clinical studies are financially supported to develop cell therapies in diverse therapeutic areas, such as primary cerebral lymphoma, or regenerative cellular therapy. Cell and gene therapy are the perfect example of a fruitful environment fostering breakthrough innovation for the benefit of patients. This dynamic is precisely what we aim to do at the Health Innovation Agency. Our goal is really to create the most attractive ecosystem for health innovation in France by coordinating and boosting all the actors working on that topic: ministries, administration, academic researchers, physicians, entrepreneurs, patients, industry. In order to do that, we need to consider innovation through its entire journey, from academic research to its effective commercialization, passing by academic research and tech transfer, clinical research, market access, and manufacturing. Our goal is really to impact each step of this journey to improve and facilitate the access of innovations to patients. Specifically for biotherapies, the government launched 2 years ago, almost to the day, a national strategy, and we celebrated its birthday on Wednesday. I would just like to salute its coordinator, Anne Jouvenceau, who is in the room here today, and she's deeply committed to this mission. So this national strategy is focused on biotherapies and biomanufacturing, aiming to position France as the European leader for biotherapy by 2030. Since its launch, France already gained 1 place in this ranking, becoming the 2nd country in Europe. This strategy contains 19 concrete actions organized around 4 pillars: education and skills, industrialization, regulation, and market access, structuring all the sector. These actions aims to act through all the value chain of biotherapy, from research with a priority research equipment program called PEPR to address the future technical challenges associated to production and development of biotherapies, to market access by leveraging all the existing early access schemes, passing through supporting tech transfer in order to transform research into new therapies available to patients. And as you can see, this strategy aims to ensure a continuous flow of breakthrough innovations by acting on each step through the value chain of health innovation. Ensuring this continuum is precisely the core mission of the Health Innovation Agency. We act at each step of the journey of an innovation, from horizon scanning to academic research, clinical research, access, and manufacturing. So first, at the very beginning of the value chain, we are currently setting up a system of horizon scanning in order to characterize the future needs of the healthcare system. We are working with academic researchers, industries, and administrations in order to embrace all the stakes, patients' needs, financial impact, organizations of healthcare. And this horizon scanning system will have a dual purpose. First, to anticipate the financial and organizational impact of such therapies. If we take an example of cell and gene therapies, these medicines have the particularity often of a single administration with a high value budget. But beyond the clinical benefit for patients, these innovations can also potentially trigger major changes in the healthcare pathway and provide long-term savings, which have to be taken into account. And we need to anticipate all these aspects in order to secure the access. Secondly, this horizon scanning will help to prioritize the government investments to support the uptake of innovation in fields where medical needs are or will be insufficiently covered, and to ensure that the entire population will benefit from them. Secondly, we aim to support the excellence of biomedical academic research by reducing the split between research and care and fostering public-private collaboration. With France 2030, we invest massively in structuring projects such as the University Hospital Institute and bioclusters, which aim to create innovations ecosystem, to attract and retain talent, and to facilitate the setup of industries. Today, France 2030 plan supports 5 bioclusters, of which Génoterre, which will enable the creation and development of dozens of companies from research to industrialization. It will attract the best talents and accelerate R&D projects. The ambition here is really to develop around 10 gene therapies for rare and common diseases. The second one is Brain and Mind, which is dedicated to neurologic and psychiatric diseases, which aims to launch rapidly product development trials with an integrated approach combining technologies, medical and non-medical tools. The third one in the field of cell and gene therapies is Paris-Saclay Cancer Cluster, which aims to change the pace of innovation in oncology to make it more predictive, more precise, and effective in the oncology field. Third, clinical research is one of the top priorities of the President Macron Health Innovation Plan for 2030. Our ambition here is to double the number of industry-promoted trials, which are the most emblematic example of public-private partnerships. In order to reinforce clinical research attractiveness, we need to accelerate the launch of clinical trials by optimizing the administrative processes and leveraging digital tools. We need also to accelerate the trials itself by promoting new methodologies of clinical research, such as in silico trials, stratification, digital twins. These methodologies represent a hope for the development of therapies in diseases for which classical trials are not able to prove efficacy, such as orphan, ultra-orphan diseases or slowly progressing diseases. Fourth, we know that in the future, healthcare will be more personalized, more preventive, more predictive, and more precise. The future therapies will combine diagnosis and care, medicines, devices, and technologies. We need to anticipate and to adapt the market access pathway for patients to benefit from this innovative treatment as soon as it is available. And this is one of our missions as well. Then, at the very end of the value chain, we act also to position France as a leader in the field of biomanufacturing. Our goal is very clear. It's to increase the number of biomedicines developed and produced in France and to reach 20 medicines produced in the country by 2030. First, we aim to modernize the industrial tool. We invest half of the amount dedicated to the biomedicines biomanufacturing strategy in pre-industrial projects and research on future bioproduction technologies through research programs, tech transfer consortium, early stage clinical studies. Secondly, we directly fund innovative manufacturing seats. We support projects that aim either to create companies' first factory or to expand or modernize existing biomanufacturing capacities. So, to conclude, I would like to emphasize the fact that France must be proud of having some very promising actors in cell and gene therapies, as you can prove it today by your presence and commitment. These innovators are involved either in the development of therapies that aim to bring hope for patients with high unmet medical need, but also developing ways, new ways to secure, increase and facilitate the manufacturing of cell and gene therapy. Thank you very much.
Thank you very much. Thank you. Maybe if you have a question for Lisalter just before lunch break, somebody? No? Okay, thanks a lot. And then it's lunch break, so please come back at 2 p.m. for session 4. Thank you.